Okay, class, in part B here we're going to cover splicing and then the addition of the poly-A RNA, then RNA export, and then we'll also look at ribosomal RNA maturation in the nucleolus. So just to start off with some splicing, so a pre-mRNA transcript is going to have basically both exons and introns, okay? And so here you can just see a couple of genes, human factor 8 and human beta globin gene, and the exons are actually shaded in red, and the introns are this light orange. And if you have trouble remembering the difference between exons and exons are express sequences, and introns are intervening sequences. Okay, so the introns actually get spliced out, and they're removed from the structure, from the mRNA. And when they do that, they actually form this structure called the Lariat, which is right here. And what occurs is you have a transesterification reaction between this A on the 3' end and a G on the 5' end, and it forms this Lariat structure, and then the messenger RNA can then be joined together. Here's the chemical structure of it, and what ends up happening is you have basically the 5' G actually attaches via this transesterification reaction with the 2' carbon on the adenine that's sort of the central A in the Lariat structure there. Okay, and then this goes off to the 3' end of the sequence. So the 5' phosphate is actually attached to the 2' ribose. Now in a cell you can actually have alternative splicing. So in this case they're just using the example of the alpha tropomyosin gene, and the exons and introns are highlighted here. And as you can see in different sort of tissues, the transcript is spliced differently. You smooth muscle, fibroblasts, brain mRNA, they're all slightly different. So, there's alternative splicing depending on the transcript and what the protein, what the specific cell needs, whether there's added amino acid sequence or whatnot. Now, when it comes to splicing, there's actually consensus splice sequence, okay? And so you have one exon, oftentimes with an AG, and then you have other sequences that are required for the intron removal, followed by a G, and when the intron's removed, you're sort of left with an AGG. Now, the splice sites can actually be somewhat degenerate, and which can lead to cryptic splicing and other things like that, which we'll see later. But let's first look at the mechanism of splicing. So splicing is pretty straightforward. It's a little confusing, but you guys shouldn't have any trouble learning this, and you should know it. And so the way it works is basically you have this U1 sNRP, small nucleolar ribonuclear protein, okay, and that binds to the 5' splice site. And then at the A, where the Lariat structure is going to form, you have the branch point binding protein, or BPP, and also U2AF. There's a small rearrangement that occurs, and the BPP and U2AF are displaced with the U2 sNRP, and then U4, U6, and U5 triple sNRP comes in, and ultimately that will displace, at least the U6 displaces the U1, and then you get another sort of rearrangement, the Lariat forms, and then ultimately the 3' end of one exon joins with the 5' end of the other exon junction, and the Lariat is actually released. And so we'll look at all of these different steps just a little bit closer right here. So the first thing that happens is the U1 in an ATP-dependent rearrangement reaction displaces, U1 is displaced with U6, okay, and you have hydrogen bonding between the nucleotides in each. And then at the 3' end, you have the branch point binding protein with the adenine where the Lariat structure is going to form, and that also undergoes an ATP-dependent rearrangement, and the branch point binding protein is displaced with U2, okay, the U2 snRP. And then in sort of the final step of the reaction, what ends up happening is you have U6 bound to one end, and that's associated with U2. It brings them together, and the transesterification reaction between the A that forms the Lariat structure and the G on the 5' end of the intron is formed, and then U5 can come in and help to join the 3' end of exon 1 with the 5' end of exon 2. So in total, this is very simplified. It actually takes on the order of 200 proteins, and there's actually proteins left behind at the exon junction, which is a good quality control to see if splicing occurred properly when the protein gets translated. And we'll look at that in a couple weeks. So poor consensus sequences and mutations can also cause alternative and cryptic splicing. So here's just a normal pre-mRNA. These yellow sequences are the introns, and normally, they'll all get spliced out. But if there's a poor consensus sequence, you can actually have exon skipping, where exon 1 is actually joined to exon 3 instead of being joined to exon 2. You can also have cryptic splice sites actually in the exons, which then lead to only a portion of an exon getting incorporated into the primary mRNA transcript. Some other proteins that are involved in splicing are SR proteins and hnRPs, or heterogeneous nuclear ribonuclear proteins. And so, here you have basically an intron. The hnRPs are bound to the intron. U1 and U2 will bring this together and splice it. The SR proteins actually bind to the exons. And also on the 3' end of the mRNA transcript, you're going to have poly-A binding proteins. And we'll see in a second how the poly-A gets added to the end of the mRNA transcript. Now, there's also different splicesomes, and I'm not going to hold you to memorize every single different spliceosome. If you know the major spliceosome, I think you guys will be fine. But in addition to the major one, there's the U12 type, and the big difference is this uses U11 and U12 instead of U1 and U2. And then you can also have trans-splicing, where two separate RNA transcripts are actually joined together in somewhat of an analogous fashion. This won't actually form a Lariat structure because these two aren't joined, but they'll actually come and form this sort of pseudo structure here. Here's basically how some of the major differences between the major and the U12 type. They just use a different U6 with different base pairing, and there's a different consensus sequence. And this is largely what the major differences are. So really all you guys need to know is basically how the major, let me jump back here, how the major RNA splicing occurs and the formation of the Lariat structure. Now, the reason splicing is so important, because if you have improper splicing, it can actually lead to disease, and it's pretty common. So if you get a mutation that's basically not in a coding sequence, it can be in the intron, what can happen is you can come up with a cryptic splice site, which will add all of this transcript on. And sometimes this can actually throw the protein out of frame, depending on where it occurs. You can actually have exon skipping, which can remove a very critical portion of the protein, and you can also get the incorporation of brand new splice sites, which once again, this can potentially throw the gene out of frame when it's being translated on the ribosome. And so all of these different pathways can occur where you can have exon skipping or cryptic splice sites or a whole new exon getting added into your mRNA transcript. You can also have self-splicing. There's group 1 and group 2 self-splicing. And these sort of just occur somewhat spontaneously. I don't typically hold you guys to knowing this exactly how this occurs. It's very similar to the other splicing. Just know that there's these sort of self-splicing can occur on transcripts. Okay, so now on to the poly A. So once the RNA polymerase has sort of reached the end of the gene, it'll hit a termination sequence, and then it travels down, a little bit past that site, and then there's a cleavage reaction where you lose sort of some of the RNA that's on the end, and then that's cleaved, and then the poly A addition protein comes in and actually starts to add a poly A sequence. Now this poly A sequence can be variable length, and believe it or not, oftentimes the length of it will determine the stability of the messenger RNA. That's one of the important things about the poly A tail on proteins, and it usually occurs after the 3' UTR. So the polymerase will travel down, you'll have this GU and U-rich sequence, and then it's cleaved. This is degraded in the nucleus, and then you get your poly A tail added to the messenger RNA. And the way that works, and I want you guys to know this and be able to explain how it happens. So when the pol2 hits a termination signal, what happens is the CTD tail of the polymerase is largely only phosphorylated on the S2. Okay, so S5 at initiation, S2 and S5 during elongation, which recruit the splicing factors, and then when it gets to the termination, it's largely S2. And then you have these proteins, CPSF and CSTF, that actually can associate with the serine 2 phosphorylated tail. They bind to the RNA transcript and actually cleave it off, and then poly A polymerase comes in and starts to add the poly A tail. And then you have poly A binding proteins that actually will bind to the poly A tail of the messenger RNA. And once again, the poly A tail is variable length, and depending on how long it is, that can actually affect the stability. And there's actually enzymes in the cytosol that will chew back the poly A tail to help degrade it, because once the poly A tail is removed, it makes the messenger RNA very unstable, and oftentimes it's degraded by the cell. Okay, so once the messenger RNA is fully processed, it's basically export ready. So let's look at this. So you have initiation where the RNA polymerase is creating the messenger RNA, the 5' cap is put on, and as the RNA polymerase is traveling down, the messenger RNA is then spliced, and eventually when it hits the termination, the RNA is then fully processed and the poly A tail is put on. And then once that's all taken place, the mRNA is export ready, and it's actually exported out of the nuclear pore complex. Okay, and this is a regulated event, and we'll look at nuclear transport in the final third part of the course. And the reason that it actually has to be exported out is no translation occurs in the nucleus. All of the translation occurs out in the cytosol. So once the transcription is complete, the mRNA is transported out of the nucleus so that translation can proceed. And we'll cover translation in the next lecture. Here's a great electron micrograph of mRNA being exported out of the nucleus. Here you can see the nuclear envelope, and then there's a nuclear pore here, and this is a messenger RNA being transported across the nuclear pore. Here's sort of a schematic of how it works. You have these EJC proteins. These are exon junction proteins. That's actually where the exons were joined together. You have the SR proteins. CBC is bound to the 5' cap, and then you have your poly A proteins, and then a nuclear export receptor will actually help the export of the protein. Once it's out in the nucleus, something very important happens where EIF4E and EIF4G actually displace the CBC and bind to the end, and it creates this sort of circular structure on the RNA for translation to occur. And we'll see this sort of circular structure when we cover translation in the next lecture. And once again, the SR proteins are actually removed prior to export. So here's just some transcription of ribosomal RNA. So here's just a piece of DNA, and here are basically ribosomal RNAs being made. And ribosomal RNA is the most abundant RNA in the cell, and there's over 10 million copies of it in a typical cell. And all of the messenger RNA, or all of the ribosomal RNA, is actually transcribed in the nucleolus of the nucleus, which we'll see in a second. And it's processed there in the nucleolus as well. So you end up producing a 45S precursor ribosomal RNA that actually gets processed and cleaved into an 18S RNA, a 5.8S RNA, and a 28S RNA. Okay, and the 5S RNA is actually made elsewhere. And these red circles and purple triangles actually represent chemical modifications that occur to the ribosomal RNA. So the ribosomal RNAs are actually getting modified. And here are the modifications. You get basically uridine gets converted to pseudouridine, and then you can actually get 2 O-methylated nucleotides on the ribose. Okay, and the RNA modifications are actually done by snoRNAs and snoRNPs. Okay, the ribonuclear proteins. So ribonuclear proteins and the snoRNAs. And the snoRNAs will actually hydrogen bond in and around where the modification is to occur, while the snoRNP actually causes the change in the base. Now, all of this is actually happening in the nucleolus of the nucleus, so this is a subdomain, and so we've seen this before, where on the outside of the nuclear, on the inside of the nuclear envelope, you're going to have your peripheral heterochromatin. Then you have this darkly stained area, which is the nucleolus, and this is where the ribosomal RNAs are being processed, not only transcribed but also processed, and it's also where ribosome assembly occurs, and we'll see that in just a second here. But the nucleolus is actually pretty dynamic, and if you guys remember back to sort of some of the karyotyping and schematics that we did like 4 lectures ago, there's actually 10 different ribosomal RNAs on the different chromosomes, okay? And they actually come together to form this nucleolus. They all sort of coagulate in the nucleus. But when meiosis happens, or mitosis, that's actually broken down as the chromosomes condense. Okay, so the nucleolus is present in G2, and then at the start of mitosis, all the chromosomes condense, and the nucleolus is broken down. The chromosomes separate, and then the nucleolus starts to come back together again, and it'll eventually, they all sort of come together and coagulate during G2 and S phase. So once again, as the cell division progresses, the nucleolus comes apart as the chromosomes condense, and then it's brought back together again. And here's sort of just an example of this. Here's the nucleolar fusion, where you have just all the different ribosomal RNAs. They're starting to come together. Here there's like 4 and maybe 5-ish in this nucleus, and then they come together, and then they sort of just all form a single nucleolus in the nucleus. Sorry. And the nucleolus is actually where the ribosomes are assembled. Okay, so here in yellow is your nucleus, and sort of shaded blue here is your nucleolus. You have transcription of the ribosomal RNA genes on the DNA, the ribosomal DNA, and that creates the 45S ribosomal RNA precursor. Those get sort of cleaved up and then modified by the snoRNAs and the snoRNPs, and then they get incorporated into the ribosome. Okay, the ribosomal RNAs help to make up the ribosome. And next term, when we look at it, the structure of the ribosome, you'll see that it's mainly RNA. There are a whole bunch of proteins associated with it, but by mass, it's mostly RNA. And so here you're starting to form the 60S and the 40S ribosomal subunits, and then they actually get exported out of the nucleus so that they can be transported. This image also shows the telomerase proteins and the telomerase RNA, which actually add the telomere sequence to the ends of the chromosome, and that's going to occur on the nuclear periphery here. Okay, you also have proteins being imported and mRNA being exported out of the nucleus. So this is just really, once again, the nucleus is actually a highly organized and dynamic structure, and you've got things coming in and things going out all of the time. And so here's just sort of more evidence of how organized the nucleus is. So panel A is stained here for fibrillin, and then you have in B here all of the splicing proteins, and then in blue you have the bulk chromatin, and you can see the splicing proteins sort of overlap with a lot of the bulk chromatin, and then you have these cagal bodies here stained in blue. Okay, so once again, the nucleus is actually a really highly organized structure when you do sort of the overlay of all those different areas. And so here's just another schematic of that. You have your nucleolus, you have your cagal bodies. We'll cover the cagal bodies later, but you have these sort of interchromatin granule clusters, and then sort of your sites of active transcription and RNA splicing, and the newly synthesized RNAs get put out, as are the snRNAs, which then become snRNPs, and they're brought back into the nucleolus, into the cagal bodies. So that's pretty much it for today. Once again, your exam is next Thursday. And for next Tuesday's class, it's all on translation. So just read Chapter 6 and the pages shown here for the 5th and 6th editions. And I will see you all on Tuesday afternoon. Okay? I'll talk to you later. Bye-bye.